developed the Varro.
He got a PhD in immunology. My PhD is in neuroscience.
The Cirrito brothers, Tom and John, shared a lot growing up together in Fort Lauderdale, Florida.
Oh, these are great! Holy moly.
And all these years later, they began sharing their science, research, and business expertise to join together on a new adventure, creating this laboratory in the Cortex Innovation District for their company, Varro Life Sciences. That's because the brothers have what no other brothers in the world have, their very own secret sauce, they call it, to detect airborne pathogen threats in real time.
The biosensor that John has developed, which we're developing at Varro, is called a microimmunoelectrode, and we call it an MIE for short. So the MIE is the biosensor that is inside, it's the secret sauce, if you will, of the breath-based diagnostic and the air biodetector.
This is the breath test for quick and accurate results.
It forces those liquid, the aerosols, through these channels to the MIE, and the MIE is here. So this is the biosensor. This is where we detect if your breath contained COVID or flu or RSV.
And Varro has a pathogen air biodetector for what's in the air that people breathe.
There is no other technology today that can monitor pathogens in indoor environments in real time. It doesn't exist.
The focus is on indoor areas, classrooms, hospitals, airports, anywhere.
The war room of the White House or the brig of an aircraft carrier or submarine, right? Any infectious diseases can have massive impacts on troop readiness, on national security.
Indoor air quality monitor, you could do it for bioterror threats, right? I mean, you could do it for anthrax.
It's a very sensitive technology that was invented by John that can detect pathogens in aerosols, which are the tiny droplets that are released when we breathe or talk in the air. So we use a special kind of antibody called a nanobody.
He had started a company on infectious disease. I had a technology for Alzheimer's that I could adapt pretty easily. So depending on how many sensors you have, you can detect that number of pathogens that would be in the air. So we can detect viruses, we can detect bacteria, we can detect fungus, all depending on what we put on the surface of that electrode. And right now we're developing something with 4 electrodes, but that number can go up, in dozens, detect different things at the exact same time.
As a scientific founder of Varro and professor of neurology at Washington University School of Medicine in St. Louis, Dr. John Cirrito helped develop the biodetection platform that he's now advancing for manufacturing and continued research in Varro's new space.
And so the manufacturing component of our operations will be in this space here, both a manufacturing facility as well as an R&D facility.
Before all of this, take a look at the beginning stages of collaboration inside the Air Lab, the interdisciplinary aerosol research lab at the McKelvey School of Engineering at Washington University. Dr. Carla Udy, professor of psychiatry, and Rajan Chakrabarty, principal investigator of the Air Lab, are part of the WashU team. The 3 researchers are the scientific founders of Varro Life Sciences. And now Varro is getting a great start in St. Louis.
Varro raised $20 million. With that $20 million, we've initiated some of the design and engineering work around a commercial device. We're building laboratories.
The company's new space is the beginning of something big.
Currently, John is taking the lead on building a library of antibodies. It's going to be very important for us from an R&D perspective. The next thing we've never seen before that threatens the next pandemic, Varro will be in a position, by leveraging this library, to produce new breath-based diagnostics and new air biodetectors extremely quickly, within 4 to 6 weeks, so that we're no longer playing catch-up with these epidemics and pandemics. Our real commitment as a company on the pandemic preparedness side is to be able to develop new devices and get them out in the field very, very rapidly, so that we can either bend the curve or hopefully even prevent the next pandemic from occurring.
That library will give us the opportunity to very rapidly create a new sensor, as opposed to what you normally have to do is go to animals and generate an antibody, which takes months to do.